Welcome back. The Academy supports the career development of early and mid-career researchers through a number of initiatives, as I was saying this morning, including workshops that some of you will be attending this afternoon and that I warmly encourage you to attend. We also celebrate the extraordinary work by early and mid-career researchers in this country with several highly contested awards, and I can tell you from sitting on one of those award committees, they are very highly contested, and we hope they continue to be so. Today we're making 10 awards that recognise the outstanding achievements of young researchers, and we'll begin with the 2014 Fenner Medal, which is made in honour of the many outstanding contributions to science by the late Professor Frank Fenner. It recognises distinguished research in biology, and my warmest congratulations to the recipient of the 2014 Fenner Medal, Professor Katherine Belov. Please come to the stage.
Thank you very much, Suzanne. Very warm congratulations. Look forward to your talk. Thank you. Oops, sorry. Medal straight away. It was my fault.
Professor Belov's research on immunity in marsupials and monotremes provides new understanding of mammalian immune systems and has great potential for managing wildlife diseases. She overturned the paradigm that Australian mammals have primitive immune systems and demonstrated that they have immune gene complements similar to our own. She discovered that it is low diversity in the major histocompatibility complex that allows the spread of the Tasmanian devil facial tumour disease, and has identified novel antimicrobial and venom peptides of potential biomedical relevance. Professor Belov, congratulations. I invite you to present to us on the topic of genomes of Australian animals, the devil's cancer, defence and new elixirs. Thank you.
Thank you, Marilyn. It's a real thrill and honour to be here. Australia is home to some amazing animals. In fact, we've got about 245 species of marsupials and 2 species of monotremes. And these 2 mammalian lineages have been on independent evolutionary trajectories from eutherian or placental mammals for about 130 million years. So there's been lots of time for these species to evolve and adapt to our unique climate. My research has focused on studying the genomes of our marsupials and monotremes in order to be able to understand their immune responses. And I've done this for 2 reasons. Firstly, to aid conservation, and secondly, to discover novel genes involved in immunity and defence. When I first started my research, my focus was looking for similarities between our own immune system and that of marsupials and monotremes. And I was able to find lots of similarities. In fact, we share most of the same genes with marsupials and monotremes, immunoglobulin genes, T-cell receptor genes, MHC genes. And by studying the genomic locations of these genes and their phylogenetic relationships, I was able to map a really nice picture of how mammalian immunity evolved. But more recently, I've turned to focusing on the differences between our immune systems. And the most obvious place to start looking is early in life, because marsupials and monotremes are born at a much earlier stage in development than human babies are. When marsupials and monotremes are born, they're tiny. They don't have any immune system, they don't have any immune tissues or organs, and their adaptive immune system develops entirely while they're in the pouch or in the burrow. And both of these environments are dirty and pathogen-laden. So I've had an interest in trying to understand how these young joeys survive in such a harsh environment. My collaborators and I have identified expansions of antimicrobial peptide genes in the genomes of these species. Now, the number of these genes, the sequence of these genes, and the activity of these peptides differs depending on which lineage you look at. What's interesting is these peptides are produced in the milk of the mother, in the lining of the pouch of the mother, and also in the skin of the young. And we think these peptides are the factors that protect the young from all the pathogens that they face while they're immunocompromised. So what we've been doing is synthesising these peptides and testing them against a range of pathogens that impact humans and other animals. And to our delight, they've been highly successful. These peptides are outperforming some existing antibiotics, and some of them are actually active against multi-drug resistant bacteria, which is very exciting, and we're really hopeful that one day we might get our antibiotics out of the pouch. We've also worked on platypus venom. Now, some of you may not be aware that male platypuses are venomous. They have a spur on their hind leg, which is connected via a duct to a venom gland. During the breeding season, this venom gland increases in volume and produces venom. Now, male platypuses don't use this venom to catch prey, rather they use it against other males. So what we think happens is the spurred platypus swims away and licks its wounds while the spurrer gets the girl. So the platypus venom doesn't kill a platypus, but it will kill a dog. And if a human is spurred, it causes excruciating pain and swelling. And it's a pain that's so severe that traditional painkillers like morphine can't control it. And victims are put under a full anaesthetic block till the symptoms go away, and that can be months. So obviously we're interested in understanding the composition of this venom because I think there's potential for novel therapeutics there. If we can understand the pain pathways that these peptides target, then perhaps we could control them and develop new painkillers. You may not be aware that echidnas also have spurs on their hind legs, the males, and they also produce a secretion during the breeding season. But there haven't actually been any cases of echidna spurring, and we don't actually think that they can erect their spurs on their own. But our interest was trying to characterise both the venom of the platypus and the echidna using the latest sequencing technologies. And to cut a very long story short, I can tell you that we found that they were very different. The echidna crural gland is essentially a scent gland, and they use their secretions for pheromone marking, whereas the platypus gland contains a treasure trove of novel peptides that we're planning to spend the next few years working through, and hopefully one day we might be able to develop a novel therapeutic in the area of pain or inflammation or blood pressure. The project that's probably been closest to my heart over the past 7 or 8 years is Tasmanian devil facial tumour disease. So this is a contagious cancer that's decimating our Tasmanian devils. It's actually not caused by a virus or a bacteria. It's a cancer cell itself that's spread by biting, and devils do this a lot. So the reason I got interested in this disease is that it doesn't make sense. How can you have a cell pass from one animal to another without invoking an immune response? We've been able to answer that question. We've known for a long time that devils have very low levels of genetic diversity. Well, we were able to show that this low diversity extends to the major histocompatibility complex. Now, MHC genes are the genes that are matched between donors and recipients for organ transplantation. And in the case of devils, MHC diversity was so low that essentially they were immunological clones. On top of that, we were able to show that the tumour cells actually downregulate cell surface MHC expression. And by using these two mechanisms, devil facial tumour disease has been able to slip under the radar of the immune system and pass from animal to animal undetected. And it's passed through about 100,000 animals now and resulted in the loss of 85% of our devils. So most of the focus in my lab at the moment is on trying to save the devil from extinction. We're working on the insurance population, managing the genetics of that population. And more recently, the question that's piqued my interest the most is why do young devils not catch DFTD? Interestingly, we would have expected local extinctions by now. That hasn't happened, and the reason that hasn't happened is that young mums tend to hang on till they've weaned their first litter. So is it just a case that young devils don't get bitten very often, or is it something to do with the uniqueness of the marsupial immune system? So we often hear that the cure for cancer can come out of the rainforest. Well, I also think that the genomes of our Australian animals also hold many secrets, and I think there's a lot of potential to develop therapeutics there that could benefit both humans and other animals. I'm very fortunate to go to work every day with the most amazing students, postdocs and colleagues. I don't have time to run through everybody today, and in fact I'm sure I've forgotten somebody important, but this really does make my job worthwhile. And I know I'm out of time, but Marilyn's on this slide. I've been really fortunate in my career that I've had the most amazing mentors guide me and advise me throughout my career, and I couldn't have done it without them. They're fantastic role models. I particularly want to thank Hugh Tyndall-Briscoe, who nominated me for this award. That's particularly important because he is, the pioneer and the grandfather of this field, so thank you. And very finally, I also wouldn't be here if it wasn't for the financial support that I've had. I particularly want to thank the ARC, who've supported me over the past 10 years, in particular the Future Fellowship, which helped me establish my career, and I'm really pleased that that fellowship scheme is continuing. The University of Sydney and the Faculty of Vet Science for being such great places to work, and to my family and friends. Thanks very much for your attention.
Thank you, Kathy. Questions? Jenny. And then Oliver. Just while we're waiting for the microphones to get there, anybody upstairs who has a question, please go to the microphone because it's extremely hard to see you and you're like in a dark haze up there. So could you please, if you have a question, move to the microphone and shout.
Jenny Graves, La Trobe University. The devil facial tumour disease makes me wonder whether the evolution of the MHC diversity, one of the drivers is the avoidance of transmissible cancer. Would you like to say something about the evolution of the MHC?
It's an interesting idea. There are only 2 contagious cancers. The other one is a sexually transmitted disease in dogs. And in that case, it evolved or it emerged in inbred populations with a lack of MHC diversity. So I think that is a trigger. In human cases, for instance, a mother can pass a cancer to a fetus. Again, there's no MHC barrier. You can have cancer transmitted through organ transplantation, and again, there isn't an MHC barrier in those cases. So it wouldn't surprise me if that was one of the reasons that the MHC did evolve.
Oliver is next.
Delightful talk, Kathy.
Thank
Oliver
you.
Mayo, CSIRO. Do you think there are any risks in using mammalian antimicrobial peptides as therapeutic agents, because you could then get resistance develop and transfer to many other mammalian species?
In this case, I've actually just had a student email me this week. She's done a lot of toxicity testing, and they're not toxic to human cells. In fact, they're not toxic to mammalian cells in general because these peptides act by punching holes in the membranes of bacteria and fungi due to a positive-negative interaction. So I don't think that's likely.
Thank you very much.
Could I just, sorry. What I was saying, you use it as a therapeutic agent and then the microbes develop immunity to it, and because of what it is, that'll mean transferable resistance in these microbes, not whether it'll cause harm in itself.
Right. I'd have to think about that. I'm not aware of any resistance to these peptides, but then they haven't been used for a long time. They're naturally, though, in all of us. We just have, say, one catholicide in our genomes, whereas the marsupials have had massive expansions of them.
I'm sorry, we're out of time again. Please join me in thanking Professor Belov.